Okay, so hello everyone. Welcome to the PHARMECO project webinar on PAT. My name is Daniel Myatt and I work as a senior analytical scientist at CPI in the UK, which is one of the project partners in the PHARMECO consortium. If you don't know, just some background to PHARMECO, the PHARMECO project looks to advance safe and sustainable quality by design practices in pharmaceutical manufacturing. One way of doing this is by the use of PAT. So today's talk is the dual role of PAT in continuous biomanufacturing, ensuring process monitoring and control while advancing sustainability. If you do have any questions while you're listening to the talk today, if you can please put them into the chat and we'll pick them up with the speakers at the end of the talk today. The talk should last from the three speakers about 45 minutes, and then at the end we'll take some questions for the speakers. So to start the talk today, I'll hand over to Brent van Geet. He's a PhD student in Professor Thomas de Beer's lab at the University of Ghent to begin the talk. So Brent, over to you.
Thank you very much, Daniel, for the introduction. As you already said, my name is Brent van Geet, and I will talk a little bit more about the dual role of PAT in continuous biomanufacturing, ensuring process monitoring and control while advancing sustainability. First of all, I go quickly through the agenda of the webinar today. First, I will go to the introduction of PAT, so what actually PAT is. Then PAT potentials for sustainability. Then I will give an overview of the different PAT tools we have available. Then the fourth point is the role of data science. I will not give this. This is by Celia, and she will also give a case study. Then after that, Joshua will take over, and he will talk a bit more about model-based applications, soft sensors, and model-based control, and also an application on PHARMECO itself. So, introduction to PAT. What is PAT? If we look at the definition, PAT stands for Process Analytical Technology. It can be defined as a system for designing, analyzing, and controlling manufacturing processes to timely measurements of critical quality attributes. So first of all, it is used to designing, analyzing, and controlling the process. And there we will take constantly time points to get the quality attributes that we want. Also, we see that PAT analytical is included there. And this is because we want to include chemical, physical, and microbiological measurements as well. So what's the goal now of PAT? The goal of PAT is to control and enhance process understanding, guided by risk management. And then in the end, we have the quality by design, real-time release testing, and continuous manufacturing, but more on that later on. So this was a definition according to the Pharmacopoeia. And if we then look more into the timeline, how PAT came into the world, let's say, it was first introduced by the regulatory agencies, the FDA, where they published PAT, a framework for innovation, pharmaceutical development, manufacturing, and quality assurance. This was 2004. 2 years later, the European Medical Agency started developing a PAT team, specifically around PAT, and they also published a reflection paper about PAT. Then later on, the International Conference of Harmonization also brought out some letters, and they first start talking about quality by design, QBD. Then a little bit later, 2010, they added a word on top of this with quality by design and development. So not only using PAT to designing a production line, but also while developing to get PAT included there as well. And then 2014, the EMA again published a guideline with PAT includes in process validation. And last but not least, in 2020, we're now talking about continuous manufacturing, real-time release testing, and digital twins. So the continuous manufacturing, so not from the batch production we have, nor continuous, but that way we have to know what the continuous line is doing constantly. So that PAT will be included there. And the real-time release testing with the continuous line, constantly releasing product and knowing what the quality of it is. For a digital twin, I will give Joshua will give more on that later on. So again, the goal of PAT is to enhance and understand and control the manufacturing process, which is consistent with our current drug quality system. So what we actually have is when we look at the process, we have the input variables and we have the output variables. And previously, we did not really know what actually was going through the process. It was like a black box. And with the PAT, we really try to get rid of this black box and really understand what the process is constantly doing. Then if we look at the QBD, when you design the process and you also have the process under control, you have the scientific knowledge and the process understanding. On all of these steps, we use PAT to really have an idea what the process constantly is doing. This is the quality by design. Then, why did we actually put PAT in PHARMECO? First and for all, in PHARMECO, with the biomanufacturing, we're using a continuous manufacturing line. And this, as I already mentioned before, PAT is really necessary to constantly know what the process is doing. But also, as it is PHARMECO, we really want to improve sustainability. We want to minimize waste and resource use, enhance process efficiency, reducing water for injection and energy by 20%, also with reducing material use, and constantly have a real-time quality assurance of our teams. Now, PAT, where can it be implemented? So some theoretical things. First, we have offline. We have like a conventional analytical test we do on a sample we took for our process stream. So if you look at the process stream, we take a sample, bring it to the lab and measure it there. That is offline. Then we also have at-line. This is where we take a sample again from our process stream. This can be manually or automatically, but the biggest difference is we sample it at the process site itself. So we don't go to the lab. This is the at-line. Then we also have the online. This is where we make a diverting portion of our production stream and we measure it on that diverting portion. We put a probe on the diverting portion and get there the measurement. This stream can then be discarded or returned, but this is the online measurement. And then we also have the more invasive one, but also the most real time is the inline, where we put the probe inside the process stream and we have a constant idea what the process is doing. Now quickly, an overview of our system for CPI. This is the continuous biomanufacturing setup they are using at CPI, where we have the upstream part, the bioreactor, and then all different downstream steps, where we start with the protein A capture, some different filtrations, then the anion exchange, the cation exchange, and again some different filtrations. These steps in mind, I will go further onto the next slide. So PAT potential for sustainability. This is where I really started, is looking at literature. What is the potential of PAT in biomanufacturing? So again, to make sure, this is according to literature. In general, I found that implementing PAT can reduce the batch failure, but it can also improve the process performance by 20% of yield. It can also reduce the quality control and the quality assurance and the labor cost by around 20%. Then if we look more closely to the bioreactor, implementing a Raman device can increase the yield by 20% and reduce media consumption. Implementing a biocapacitance probe can increase the success rate by 2%, and a 2D fluorescence and mid-infrared can also reduce media consumption. Then if we look more closely to PAT chromatography, protein A chromatography, at-line HPLC, can constantly track the product concentrations in loading and elution streams. Also, it increases the resin capacity by 25%. This makes sure that we get more product purification in each step, each cycle, sorry. It can also reduce the buffer consumption by 20%. Now, in the previous slide, I talked about all these different steps, and now I'm only showing two. You could ask, why are you doing that? That is the biggest reason is when I also looked at literature, we first want to know what is the least sustainable step in the whole continuous line. And if we looked at literature, we saw that the bioreactor and the protein A chromatography were by far the least sustainable. This according to energy consumption and water usage. So that's why we now in PHARMECO first start with those two steps, getting those ones more sustainable because they are the least sustainable, the biggest portion to aim. So now, the overview of different PAT tools. First and for all, we have the spectroscopic methods. We have the infrared, the UV-Vis, the X-ray fluorescence, the NIR, and the Raman. We also have other ones, but I'm just gonna, for now, go talk about these ones. First and for all, the infrared can measure nutrient profiles, water and media consumption. UV-Vis can measure cell debris, protein concentrations. X-ray fluorescence can measure element impurities, media minerals compositions. NIR can also measure metabolites, glucose, lactose, glycerin, and all different things. And last but not least, the Raman. It can measure a lot of different things, but I'm not going to go further into the Raman because that is for Celia. If we look more closely to the upstream part, we can implement a pressurized CO2 where we measure the CO2 accumulation. We can also add a weighing cell, mass flow. This is just basically a balance underneath our bioreactor. And this way we can measure the media composition and the flow, the feed rate of our line. We can also measure use capacitance probe to have an idea about the viable cell volume. Also, gas analyzers can be used. To the refluorescence, as I said before. Tailor dispersion, it can measure large aggregates and protein titer, and particle size analyzers. We can measure cell aggregate size, bead, particle distribution, things like that. Then for the downstream, we have again all our different, not all, but a lot of different spectroscopy methods. But you can also use HPLC, HPLC-MS, which can look then at protein purity, charge variations, and also PTMs like glucosylations, oxidations. And also we have the variable PET length spectroscopy, which can look at a wide range of protein concentrations. So now, as I said, we can use spectroscopy, but when you have a spectrum, we don't yet know how much glucose is inside the bioreactor. So that's where the role of data science in PAT is very important. So I'm not going to talk further into the data science itself. This is for Celia, and then thank you for listening already.
Thank you, Brent, for this transition and introduction. So yes, indeed, when we speak about data, we need different tools in order to be able to analyze them. And this is what we will see in a bit detail just after that. I'll take an example of the use of Raman spectroscopy in order to make it more concrete. So as a first step, I'll just do a little introduction about what is Raman spectroscopy and how does the Raman effect work. So first, what is the Raman effect? So it's a nonlinear process consisting of the exchange of energy between a light source, for example, from a laser, and the vibration within a material within the chemical bonds in a molecule. If we take the example here of the molecule of water. So there is different type of vibration, like you can see on the animated GIF here, different types of vibration. When the light source interacts with those bonds and those vibration, it results in different change of energy, and it, as a consequence, there is a change of the color of the lights, and it has a different Raman effect for each type of vibration. As a result, we can have like a spectrum like this for each molecule, and really it's really correlated to the type of chemical bond and vibration of those bonds. So the Raman spectrum is like a molecular fingerprint. So this is the spectra of the water. If we have a look, for example, at the cyclohexane molecule, the spectra of the cyclohexane, we see that, so here we analyze an organic compound that is pure in this media. So we have very thin bands, really easy to define. So this is easy to analyze. But when we have a look at more complex medium, such as cell culture medium, we have a lot of different molecules present in the media, in the what we want to analyze, and as a result, the spectra is more complex with a lot of different signal overlapping on top of each other, and it's hard to define what do we have in this media. And this is where we need the chemometric analysis in order to decorrelate all those compounds and be able to have a link between the spectra in the one hand and the concentration of what we want to analyze in the other hand. So this is why we use chemometrics. Now, so yeah, we do need chemometrics to extract the relevant information so that we can understand our process. Now, what is chemometrics? So if we have a look at the word, in two words, chemo for the chemicals and metrics for the measurement, how to measure the chemicals. In the field of data analytics and of data in general, we hear a lot of different buzzwords. Now let's try to have a better view of what we are speaking about. We have the big scope of artificial intelligences. Within this field, we have the machine learning, which is composed of different methods and linear and nonlinear methods. And within the field of machine learning, there is the chemometrics, which is mainly composed of linear data analysis methods. And this is what we want to use for our analysis here. So a first tool that we use a lot in this kind of analysis is the principal component analysis. So it allows to have a qualitative measurements and to explore multivariate datasets. It allows to identify outliers and identify different groups, clusters, or trends. For example, if we have a look at the graph here on this slide, on the right of the slide, we have a score plot of a principal component analysis. What we have to take into account is that each dot represents one spectra recorded during a process, and each color, if we have 5 colors, different colors, represents different batches. So the spectra were collected through the batches, and each color is a different batch. And just having a look at this figure, we are capable to see that there is one here, the light blue one, that behaves differently than the other one. So this is a tool that we can use as qualitative analysis of the data that we have to make it more simple to analyze. It can be applied on spectra or other data from multi-dimensional data sets where we gather a lot of different information. And we can use this tool, the principal component analysis, on any size of dataset from medium to very large. So this is a tool that can be used for qualitative analysis. Now, as mentioned by Brent earlier, what we want to do is to have quantitative measurement. So for that, we will use, in this example, the partial least square regression, which is the mostly used in the chemometrics as a quantitative method. So how do we do that? First, we have the part for the multivariate calibration. In the example of the Raman spectroscopy, we have our process here with a probe plugged into, for example, a bioreactor, where we have our Raman analyzer, which collects, in the one hand, it collects the spectra from the media. On the other hand, in the lab, we'll take some sample, do our offline or at-line measurements in order to determine the concentration of the analytes we want to monitor. Let's say glucose concentration, viable cell density, lactate concentration, whatever. Based on both those spectra and those reference measurements that we've done, we build a PLS model. Then there is the monitoring phase. Once the model is built, now what we only have to do is collect the spectra, and then through the software, the spectra are going through the model, and we are capable to have a real-time quantitation of what is in our media. So here, this was the different tools that we use. Now that we have access to real-time data, how do we use it? So here we'll deep dive into a case study where we use those real-time value from the Raman spectroscopy in order to perform a feedback control loop for the glucose on the glucose concentration. So here we have our system with a bioreactor system with the vessel here and the probe plugged into the media directly connected to the computer. Once the Raman spectra is read by the analyzer, it's communicated to the software, which calculates the glucose concentration. The glucose concentration is then communicated to the bioreactor system, which, accordingly to a function that I will detail later, activates a pump consisting of a complex feed where there is glucose in it. So it was a full feed with all the nutrient for the cell culture, which had glucose, and based on the glucose concentration, the pump was adding more or less of this feed in order to have an optimization of the use of the nutrient for the cell culture run. So quite easy. How do we implement that? Also quite easy. What we have to do is to make those two systems communicate between each other. The two systems can communicate through OPC UA. What we have to do is just simply as enter the OPC address in the bioreactor software. And if we have a look at the interface, the mock-up of the interface of the bioreactor software, we have all the sensors and the hardware pieces that we have on the bioreactor software. And doing that, doing this connection, it's now possible to have also external sensors connected to it, and we can connect the Raman, and now we have access to, for example, the glucose concentration as any other parameters in the bioreactor software, at the same level of temperature, GO, whatever. Same level as any other sensors. So now what we just have to do is to set the pump rate as a function of this glucose concentration. So here was the formula that we used. So the pump rate was really a function of a glucose concentration, and each time we had a new value for the glucose concentration, the pump rate adapted automatically to this new value. So what happened in the lab? So if you have a look at the graph here, or we can see the evolution of the glucose concentration in green, the viable cell density in blue, and the lactate in dark blue. And the line represents the value that we were measuring in real time by the Raman. And the small crosses you can see are measures that we did on some samples just to make sure how accurate were the Raman measurements. And we can see that in all the cases we had really good monitoring using the Raman. But if we have a focus on the glucose concentration, we see that we were capable to have a really stable concentration with this use of the feedback control loop. At some point, we had to end the feedback control loop, and we see that the cells still continued to consume the glucose. So this very steady state of glucose concentration is really linked to this feedback control loop, and we were capable to have something really, really stable and accurate. That's the most important. So yeah, that's an example of how to use PAT and data that we have in real time in the lab in order to improve the daily life, let's say, and the processes in the lab. Now I'll let the floor to Joshua for the other type of use of the data in real time and the modeling.
All right, thank you, Celia, for the introduction and the nice talk about chemometrics and use of chemometrics also within a bioreactor and within a feedback control loop. Just a few words about myself. My name is Joshua Böhm. I'm a senior scientist at Research Center Pharmaceutical Engineering, or short RCPE. We are based in Austria, in Graz. And I'm there working in the field of process modeling and process control. And very happy to be part of the PHARMECO project here in this work package 4, where we are diving into sustainability improvements for the biomanufacturing production line. Just one quick reminder, if you have any questions on the way, don't hesitate putting them into the chat. And now diving into the topic of model-based applications, where we are contrasting a bit from the chemometrics that have been used before, which are also kind of model-based applications to get inside the PAT approach to some meaningful data. We now assume that we have PAT data in place and have a bit of look what to do with these data. And first of all, if we talk about model-based applications, we need to talk about which kind of model we are thinking of to distinguish a bit there from what we are using here and what is maybe known in general. Just one thing there, I think Brent mentioned it before. I'm talking about digital twins. To make it a bit more specific, we are talking about process models here in this context, so that we have the same understanding what we are talking about here, where we try to describe the process, which has inputs, our process settings, so our process parameters coming in, like flow rates, stirring speeds, temperatures, where we have outputs, which are, for example, our concentrations that we measure via PAT. And we have additional outputs that we know, okay, what is actually happening in the machine from the machine data that are provided by the manufacturer of the machine. What do we want to achieve with these kind of models? We want to achieve in the end that we can analyze some key metrics of the process, or that we can predict performance inside the selected process. So really to know, okay, what is going on in the process in a virtual environment. And therefore, we need to develop a model that has certain aspects. And the first thing is we want to have it in the virtual environment. Usually, if we have process data, if we know what is coming out or what we want to have there, we need some time dynamic dependencies included there. And I think this is the main difference to all the chemometric part, that there we usually have stationary models looking at one time point, and here we use the time scale as well to get to some results. The this model should then be computationally efficient and giving us sufficient accuracy. What do we mean by that? It needs to be computationally efficient to be used in real-time applications. In the end, that's something we come to in the next parts with the soft sensors and model-based control. We need to use the model in these settings, and it needs to be sufficiently in accuracy. It doesn't mean it needs to be 100% accurate at all times. It just means that, for example, if we use it for process control, it needs to be accurate enough to give us the right trends and the right magnitudes that we know what is happening next in the process. And to get to such a model, reasonable modeling approaches should be used. Probably most of you heard terms like mechanistic models or data-driven models. In the end, it's depending which data are available. If there's a lot of PET measurements available from previous experiments, then data-driven models get easier to do. Since data are there, if there are no data available, different kinds of modeling approaches need to be used. In the context of biopharma, where we are in this work package within the PHARMECO project, this model should therefore give us some kind of a virtual representation of selected process units. As we heard from Celia before, she was talking a lot about using the ramen in the bioreactor. So this would be, for example, the process would be the bioreactor to really see what is going on inside the bioreactor by means of the process model. Other units, as Brent mentioned it before, like the chromatography units, are quite consuming in terms of resources. So this is something we will certainly look into the biopharmaceutical manufacturing line here. And in the end, for these units, we want to really have a good mathematical understanding then of the relationship between the critical process parameters, the material attributes, the quality attributes, as well as sustainability metrics, which define us how sustainable we are inside the process. So going from there, now to the connection, you are in a webinar regarding PAT. So how do process models relate to PAT? And looking there at the two use cases that we have, the soft sensors and the model-based control. Starting off here, what we want to achieve by each of this with the soft sensors, we want to gather process data of the process. So we want to have more process insight. And for the process control, we want to use available process data that are gathered within a process, for example, with PAT measurements, with Raman measurements, or different methods, and use these within the process. Both should be done in real time. And the information that we want to gather by soft sensors are essentially the information that either cannot be measured in real time or cannot be measured with suitable effort in real time. So if it's really expensive, for example, to measure these, then there's a trend to the soft sensors. If we have a look at the model-based process control, what we want to achieve here is to make some decisions over adjustments in the process parameters that we use in our processes to achieve desired key performance metrics. Again, if we are talking here in this context, for example, to lower the water or the energy consumption that is used here. And the last part here, if we talk about the soft sensors, what we want here is essentially to use process models and PET measurements to gather further data of information that we don't get by the PET, to have data that is similar to some PET measurements. And for the model-based control, we want to build on these data measured by hard sensors or by soft sensors and build on these data. In the next part, I will go first into the soft sensor part, into the soft sensors, explain a bit more what do we want to achieve here, and starting off with a definition here. A soft sensor is a software-based algorithm that estimates process variables, such as the critical quality attributes, that cannot be captured by suitable hardware-based instruments. So what we see here essentially is that hardware-based PET does not work here. To get these data, then it makes sense to go for soft sensors. And if we have a look at our process again, we can split off this PET part here into, on the one hand, the variables that we can directly measure by hardware-based PET technologies that are given. For example, the glucose measurement, for sure, we wouldn't need a soft sensor for that. If we can also use the Raman prosthetics device that has been presented before. But if there are other soft sensors of interest that we cannot measure because there are limitations, then the soft sensors are needed. Limitations can be that there is no suitable measurement technique, so that there is a physical working principle that cannot be captured, that there are physical limitations, for example, in the sense of there's no space or no possibility to enter with some probe, or that it's really associated with really high cost to get a hardware-based measurement in there. So if any of this is there, then soft sensors make sense. And if we have now a look not at the process itself, but what we want to have for a soft sensor, we see here the soft sensor and we see some requirements here that we want to have real-time process data, which are on the one hand the process settings that we have, so the process parameters that we choose, actual measurements on the process data that is given, so which process parameters are actually there, and further measurements by the machinery, and also the use of directly measured variables by means of hardware-based PET. So for example, we would use here in a bioreactor to gather further information about variables that we cannot measure. We would use, for example, the glucose measurement of the Raman device here, and then from in combination with the process settings that we have, get from there to other variables that we can maybe not measure, such as some impurities that might occur here. And if we have this in place, we somehow need this translation inside the soft sensor. This is done by a process model. As I said before, it's a mathematical representation that describes somehow this transition here and describes the process inside, to say basically from the process settings that we have, how do the variables that we want to achieve, how do these evolve? And we can use further information here inside to make the estimation of these process variables more accurate. Coming from this application where we get a better picture of the process, where we have a better insight of the process, to the application of model-based control, where we now want to use the information to make something out of that. And starting also off again with some definition here of the model-based controller. This is in the end a software-based algorithm that calculates the set points for the process parameters, so the process parameters that we want to achieve, that are required to achieve optimal process performance, while considering aspects such as the quality, the sustainability, but also economical effects. So these need to hold while achieving them to get the optimal process performance, and we can adjust there the process parameters. And starting off again with our process that we have here now with both variables measured. If we go for the control, if we go here for the control, we also need some requirements. Again, we need, of course, these measurements available for achieving really the optimal control. We need the process models available, and then we can go and do something with the controller. And we need to know our objectives to do something then with the controller. In a simple sense, in the first way, feed-forward information only. We just know our objectives. We have to then calculate based on the objectives the desired process settings, the desired process parameters that we need here in order to get the best possible output, so the process performance is at the nominal optimal set point. If we do it like this, we do not need to feed back any information, but we don't have any guarantee that we actually end up at the optimal process performance. And in order to have this guarantee in place, what we usually do in process control is then to have a feedback of these variables that we either measured by PET measurements or by soft sensors, that we feed these information back inside the controller and see, well, which objectives we have, how far are we off these objectives, and calculate then based on the controller the process parameters that we want to have. Now we can have really simple controllers inside, such as PID controllers, where we just calculate the error of one variable of interest and decide based on that for one process parameter, for example, the flow rate, how we want to change it to get the error between like the objective and the actually measured variable to as small value as possible. But we can also go one step further and have the model-based calculations inside there from the process models to really guarantee to achieve the optimal process settings. Just to give you again an example here, what we mean by that. If you take the Raman glucose loop that we had before, we had one variable that we measured to have the best nutrition consumption. So we have the objective to have the nutrition consumption as good as possible. We have the measured glucose value inside the bioreactor, and based on that we calculated one flow rate. And we can combine this now to measure more information also about the product inside the bioreactor or impurities that are there, and have as objectives in the PHARMECO content now not only to optimize the consumption of one feed, but to optimize the total efficiency given by the flow rates and by the energy consumption. And if we have all this information there, the calculation becomes more complex, and then we use these models to predict into the future what happens if I change my process parameters from one set point to the other set point. How does this influence the process behavior and our predicted outcomes? And we do this until we really reach like the optimal point in terms of the process performance that we want to achieve here. Having these in place, one outlook to how we apply these things in the PHARMECO project. And starting off here again with the production line, the biopharmaceutical production line that Brent presented before, where we have the different units, the bioreactor, filtration, chromatography unit, up to the end where we have the product there. We have in each unit some product inputs, some side streams that are coming in. We have some waste streams coming out, and the process parameters that we need to set. And for the purpose of PAT and process control, we have this production line fixed in place. And what we want to do in PHARMECO is to maximize the process efficiency, which we can measure by PAT and soft sensors. And this is essentially combined out of the three things of the energy consumption that we want to minimize, of the water consumption, and the waste production, which we both want to minimize inside this production line. And thereby we can maximize the efficiency, and we want to achieve that while ensuring, of course, that the product quality is still within specifications. To get to this point by means of active process control, then we build controllers that maximize the efficiency while adjusting our process parameters in every unit in real time. We use the information measured, adjust the process parameters, and thereby get the get the real-time feedback included, get the efficiency maximized. And this can be done as shown before for single unit operations, example given focusing on the bioreactor only, but it can also be done in a way that we make this optimization over an integrated production line consisting of multiple units to have a plant-wide optimization of the efficiency and using really interdependent parameters here. With that, I want to hand over back to Brent for the summary. Thank you.
Thank you, Joscha. So now the summary of the PAT webinar. PAT can enhance the process understanding and strengthens manufacturing controls. It also enables continuous and more robust manufacturing. Spectroscopy and real-time monitoring are essential enablers for this. Chemometrics, advanced analytics, and digital twins amplifies the PAT potential. And with our PAT, we really want to try to hit the objectives of PHARMECO, where we will collaborate into development of an intensified product platform reduce the water for injection and energy consumption by 20%. We want to try to hit these objectives. So overall, PAT supports smarter, more sustainable, and a greener bioprocessing. So we'll now go into your questions. I give the word to Daniel to ask them.
Thanks, Brent. Thanks to the three speakers for really interesting talks. As Brent says, we'll now look to answer the questions that have been put into the chat. So I think the first question was for you, Brent. So Pascal was asking about slide 12, and you put on there that Raman can increase the yield by 20%. So if you can just explain a bit more.
So yes, in slide 12, I was had some numbers. This was according to literature. So in the beginning of the PHARMECO project, we started with a literature study, and to get an idea of what the potential was. And there I came one of the articles saying that it can help by 20%. So the exact numbers from there. But as Celia also talked about, her part of the presentation where using the Raman really increased the yield, let's say. But the exact numbers are not available from that part.
Yeah, thanks, Brent. Yeah, I think Celia, so in your part of the talk, so Nandita has asked what was the frequency of the Raman measurement? I assume it's like the wavelength.
Yeah, so the frequency, so for each measurement, we took 30 spectra of 30 seconds, so resulting in 15 minutes of acquisition per measurement. And we decided to have the frequency to have one new measurement every 30 minutes. So we had a new glucose concentration every 30 minutes. This was our choice. We could have something longer or shorter, but since our process didn't change that much in 1 hour, we decided to have a new measurement every half an hour, so that the pump rate then was adjusted accordingly every half an hour based on the measurement.
Yeah, okay, thanks. And then so Williams put a question. He's asking about, I don't know who the best person to answer, maybe Yosha or Celia. So the carbon footprint of training the model for the PAT compared to an offline HPLC. So he's just saying when he looked at it in small molecule manufacturing, there's a big sort of carbon footprint. So I don't know. Has anyone looked at that, the kind of carbon footprint for generating the models, or?
Yeah, I mean, like I think in general, if you have like modeling, the most or one of the largest issues is really to get to enough data, and that's really largely depending on the type of model that you use. Especially if you use data-driven models, then you have the point, and I think it doesn't really matter whether you're talking about soft sensors here or about chemometrics. If you use data-driven models, in the end, you need a lot of data to really ensure that you know how to translate this kind of spectra or data or process data that you measure to the correct concentration, for example. Think on the process modeling side, that's really the thing. You have options such as mechanistic models where you can go for modeling options with lower data, and there is a trend to really use models or try to develop models with as low effort as possible. But I think the one thing that is there on the process modeling side, at least, is the option or that there's a trend really looking into how can we reuse models, how can we retrain models if like the process has changed a bit, so that we don't need to start from gathering all data in some measurements all the time if we go to some other process. I think, Celia, that you are doing kind of similar things in the in your group as well, that you try to that you have some database, but I think that you use to basically just fine tune to the process that you're in. But I think you can talk more about that one.
Yeah, exactly. That was how I had in mind. It's we also can be like, in a way, sustainable about data directly, reuse data. In most cases, we already have a lot of data. We collect samples from historical batches, and we do have already information about that. And this is something we can use. So also be like reuse data that we already have. And also, I'll speak about what I know. Our Procelix Raman analyzer. We also have like ready to use models that already for the users to implement directly. So the data have been done internally, and now it can be deployed at on any sites. No need of new training from scratch or whatever of the models of the PAT itself. So the data is here, and there is no need for more training or not more a lot of analysis in order to be able to have a real-time monitoring. And like you just said, Joshua, it's just it can be fine-tuned using new data, but now that we have invested a certain amount of time and resources to generate these libraries, this base of knowledge, now it can be used without generating more a bigger carbon footprint for the next steps. So there was this phase of investment, but now it's cheaper in the sustainability as point of view, I would say.
Yeah, thanks, Celia. Yeah, that's a really good explanation from both of you. So John's asking about AI as well. So what does AI mean for the future of PAT? I don't know if anyone wants to take that on. I suppose it's different kind of models, machine learning and things like that. Yeah.
I can start on that a bit, because I think it's actually quite connected to the question before, with reducing like the effort to get the model there. Essentially, depending on which kind of modeling technique you use in the end, you can use the AI really to get these estimations, these transitions from one product to another product, or from one bioreactor to another bioreactor, for example, to use AI there to estimate the parameters quite quickly. I think this is one part in terms of process modeling where you can use it. I think there's a big risk in terms of AI if you want to use the total process model as AI, speaking about the process model, since then you don't have any guarantee that you stay within the physical bounds, where usually some mechanistic structure in the upper and the top layer of the model ensures that you stay within physical bounds, and using then AI to focus on the parameters that are hard to gather otherwise, that's something that's certainly helpful in terms of the process modeling. Think like where does AI really start? Where does machine learning end off? I think chemometrics is essentially machine learning. So where do you start then using the term of AI? It's some kind of learning algorithm always in there. Talking about that, I don't know whether any of you, Celia or Brent, want to add anything. In PAT directly, I think like in process modeling is really the thing. It's a really helpful, strong tool if you know, okay, this parameter is too hard to get. Then we go there and can use some learning algorithms to get there. It's a helpful tool to, once you have one process model, to learn how the parameters inside the model adapt. I think these are the two main things where you really can do something really helpful in terms of process modeling.
Okay, Cheers, Joshua. Yeah, that's a really good answer. So just on slide 11, Pascal's asking about sort of the QC, QA efforts. Can they be reduced? And with this kind of goal of the real-time release-based testing based on the data that's incoming in real time, that helps to reduce like the QC, QA involvement. So the analytical testing there. Has anyone got any thoughts on that or something on for the slide 11? I suppose Brent maybe.
Yeah, quick look at the question. So by implementing the PAT, by constantly knowing what the process is doing, we have in the point of time more an idea what the quality will be, and that way we can reduce the quality control, because in the whole time point of the process we really know what it is doing and know the quality, let's say.
Yeah. So I'm just going to do one final question just to sort of wrap up. So Joshua, what are the sort of savings that are anticipated in the kind of PHARMECO project?
Yes, if you just go there, I think in general in this work package here, talking about with a biopharmaceutical manufacturing line, one thing is actually on the slide here that we have like an anticipated water consumption by 20%, but since sustainability in the manufacturing process is not only related to water and the PMI, that is a widely used measure, but it's consisting of multiple parts. Like in the processing, we want to reduce the water consumption by 20% while simultaneously reducing the energy consumption also by 20 to 50%. And in the MIA, we've been reducing like the input material simultaneously. We also reduce the waste streams that are coming up there. So I think 20% water consumption and around 20 to 50% in this area. Then energy consumption, this combined is the goal in this work package here.
Okay, brilliant. Yeah, thanks for that. So I think we can end it there. So just thank to the speakers and everyone for attending. If there are any questions that we've missed and you want to reach out, I'm sure we can do that offline. And there will be more PHARMECO webinars in the future, so I'd look out for them if it's of interest. And thanks to everyone for attending, and have a good rest of the day. Thank you.
Thank you, everyone. Have a good day.